Hi, everyone. I am, as you had said, I'm a microbial ecologist. It's really a pleasure to be here today. And today I'm going to be talking to you about one of the branches of my research that I think really integrates ecology right into data stewardship, community-driven actions, and the need for equitability in biodiversity research and how important this is. So, so I study microbial communities, which are an interesting thing to study because they are the oldest in the world. Bacteria came first, right? But at the same time, to us, they're the newest. So up until about, let's say 30 years ago, our knowledge of the bacterial world was extremely limited. And what we used to think of bacteria was when bacteria cause disease, and really not much otherwise. With sequencing, we've been able to really see that bacteria are everywhere. They're also not just the reason that we're sick, but also the reason that we're healthy. They are the reason that soils are fertile. They're the reason that carbons cycle through terrestrial and aquatic ecosystems. So we've really come to have a much better understanding and a new appreciation for bacteria. And it's all because of sequencing. Most bacteria occur in communities, so they're very hard to take apart, which makes taking them and studying them in isolation nearly impossible. And here's where sequencing has come to the rescue, because it allows us to take a community basically at whatever time it's sampled, and with a snapshot approach, take all the DNA that's present, look at which bacteria owned that DNA, and have a very good idea of what community was there at the time of the sampling. And what's interesting is that microbial ecology is a field that has developed relatively late because we have relied so much on sequencing. So we've experienced, and we are experiencing, a revolution in our understanding of microbes at the same time as sequencing technologies are being revolutionized. And these two things go together very, very tightly. Another thing that's very interesting is that because we are a relatively old discipline, archiving of data, computers were already present when sequencing started to happen. Archiving of data was understood to be important at the outset. And so we have pretty old sequence archives dating back to the 80s. And we, at this point in time, really all know that we must archive our data, our sequence data in its raw format at the very least, in a set of very defined public archives, before we even publish our data. And so this is something that is now pretty established. And so fairness in our field is something that has emerged together with the field, which is, I think, a difference from other areas of biodiversity research where, the recording of biodiversity goes back centuries and we don't really have standards. We are a field that emerged together with these basic standards. The archives that I was mentioning are these three that you see here: the DDBJ in Japan, NCBI in the US, and ENA-EBI in Europe. And they speak to each other. They are the International Nucleotide Sequence Database collaboration. And so for us, it's actually very obvious where we should deposit data, how we should be depositing it, and there are clear disciplinary standards. The problem is that we seldom think about the reuse of these data. So these archives have been developed with the idea of saving the data as a resource, but the resource part of the saving the data has received less attention. And we haven't really started to think about, okay, we've created this resource. It's been 30 years of archiving data. These archives are super rich now. Who uses them and how? And what we've come to start to see is that actually you need computational power, you need training to be able to use the data in these archives. And this means that not everyone gets to use these archives, even though everyone has the mandate to archive in them. And so this leads to a lack of equitability. So what you see here is the Earth Microbiome Project in the map. This is the largest compendium of microbiome data that was sequenced all at once. And you can see that it's highly biased to the northern hemisphere. It's highly biased towards richer countries according to the OECD classification. And if you look on the left, you'll see that actually it's mostly high-income countries that are doing biodiversity synthesis. And one thing to note here is that on the left panel, we're not looking at microbial ecology, we're looking at ecological syntheses in general. So I would want to argue for this audience that this is something that we should all be concerned about, the fact that we are archiving data and actually half of the world is not really using it. And so this creates problems, but it creates opportunities. For me, it has been very obvious from the beginning that reusing sequence data, if you are not very rich laboratory, is the way to go, right? Sequence data is very expensive to produce. Large-scale projects like the one you see here cost millions and millions of dollars or euro. But the data that has been created by a bunch of different laboratories over the past 3 decades is freely available. And so these data sets do create the opportunity for large-scale ecological research at no cost. They also serve as a resource for training, right? If we are training people in bioinformatics and statistics, we need the sequence data to start that, and there's a wealth of sequence data available. And I think that it's also really important to consider that when only a certain part of the world does synthesis research, the conclusions that are drawn from that research are skewed, and the cases that are explored by the synthesis research are also skewed. So a very simple example of this is when there is a synthesis on the effect of agricultural inputs on the microbiome, and it is biased towards the northern hemisphere. Well, the types of agricultural inputs in Europe are very tightly regulated. And to some extent also in the US, there's less regulation in other areas of the world. There's particularly less regulation in the southern hemisphere in many countries. And so this means that we are systematically excluding the impact of a whole class of agrochemical inputs on the microbiome. Then these evade policy, and then they don't get regulated. So it creates kind of a feedback loop. So we really do need to include everyone when we think about synthesizing data. And then there's the aspect that there's really not much to be gained from archiving data when you do not get to reuse it. But what happens so this is my hypothesis to a certain extent. What happens is, for us, we actually do need to provide an accession number to the archived data before we can either submit or get our papers accepted. This is field-wide. And so what ends up happening is everyone needs to archive their data. Everyone does archive their data, but we don't do a very good job of doing that. So the extent to which that is intentional or not is something I cannot comment on. But I can say that there's not a lot of incentives to do it properly when you are unable to use the data yourself. So what you can see here is an analysis that we did. I'm going to wait for this truck to pass. Okay, so big truck. So what we see here is an analysis that we did. It's a very large scale analysis of the sequence data archives. We considered 30,000 studies. What you see here are the 2,000 studies for which we found the exact type of data that we wanted to focus on. And you can see how data is lost at every step of the archiving process. So some of the data is not located on INSDC databases. Some of that is not made public, so there is an accession number, but if you type that accession number on the portal, you will not get the data set. There's a surprisingly high rate of incorrect accession numbers in the literature. And then there's all sorts of file corruptions that can happen. And in the end, this section here, labels, is something that we really do attribute to lack of clarity in information on how to archive. And when you talk about labels, part of the problem you can see here. The metadata that people enter into these archives is extremely diverse and it can be extremely vague. So if we're talking about microbiomes can come from really anywhere, but we get really broad metadata like metagenome or gut, but we don't really know whose gut. Or aquatic, but we don't know if it's freshwater or seawater. Skin, again, no species. Human, not really specifying what kind of sample it was. So this is really a big problem. And what we have come to believe, and what I've come to believe over time, is that this is not necessarily done maliciously, right? But there really are no incentives for it to be done properly, because people don't think about the second life that these data sets get when they are reused. And so archiving has very little value to these researchers. It is just another bureaucratic step. And so what we need is to encourage synthesis research through training. I think it's really important to lower the barriers for others to do synthesis research, for the barriers to be as low as possible, so that at any point a data producer who is archiving the data can simultaneously see themselves as using that data in the future. And so the process of archiving data becomes not just a duty, but a way for them to safeguard their future potential use of the data. And that, I think, is a way that can really lead to improving the quality of archive data and metadata. And the other problem here is that the INSDC databases originally were not produced or created with credit systems in mind. So oftentimes, or in general, the data producer gets absolutely no credit for their accession number being used. Oftentimes the DOI of the publication where those data sets were originally featured gets completely lost as well. And so even if I wanted to, as a data reuser, if I wanted to credit the original creators of these data, it's very hard for me to do so. We found that approximately 60% of the data sets on INSDC don't have links to DOIs. So this is a very high rate of incompleteness. And this really makes it impossible to credit data archivers, data producers. And this is kind of all of the thinking that went into MiCoDa. So MiCoDa is the Microbial Community Database. The Sankey diagram that I showed you a few slides ago was the original motivation. So we had looked at 30,000 articles, selected 2,000 that had data types that we were interested in. And then of those, we found 252 articles that were well archived with sufficient metadata and sequenced the same region of a primer, which is really important for us for basically chemical reasons. So primers have different affinities for different regions of the same marker gene. So we really want to make sure that we're always targeting the same region. And that's what we found these 252 articles had done. And then, we got 16,000 samples from those 252 articles. We collected technical metadata manually. The idea here is to make it easy also for ecologists in their synthesis studies to have, random effects in their hierarchical models or whatnot. And we integrated these data with the Earth Microbiome Project, which had also sequenced that same region, which is the blue region that you see in the gene on the left. And this led to MiCoDa. So you can see, I showed you previously the Earth Microbiome Project data set. I told you it was the largest compendium of data. Those are now the purple dots, and you can see how much MiCoDa has enriched this data set, not just in terms of the number of samples, but also importantly, the location of samples. So a very important aspect is that the Earth Microbiome Project was done through scientific networks. And so geographically, it was quite limited because scientific networks can be geographically limited. But because what we did was just go randomly through the literature, we didn't have that specific bias. And so we could collect a lot more data from more diverse regions of the world. Another aspect of this is that MiCoDa was designed with equitability in mind. and not just equitability north-south, but we also want different types of users to be able to access the data. So I've told you previously that INSDC databases archive raw sequence data. These are large files of A's, B's, A's, C's, G's, and T's, and they are unusable for someone without bioinformatic training. They are unwieldy, and they don't look anything like the normal species by environment matrices that ecologists are used to working with. It takes bioinformatics processing to get the data to that point. And it also takes a lot of molecular know-how in order to choose the right parameters to do this. And if you are a synthesis ecologist, this is not something that you have training on, and probably not something you want to get training on in order to include bacteria in your studies. And this really does result in ecological syntheses systematically excluding microbes simply because the data is not accessible. So what we've done with MiCoDa is already done the processing. So what the user can do here is just, this is the main landing page of MiCoDa. It is already accessing the database. Users can live filter the data sets and choose what kind of samples they're looking for, and then download the data already in the sample by environment or sample by species matrix that is familiar to most biodiversity researchers. And this means also that if you do not have access to the computational power that it would take to process all of these samples, even if you do have the bioinformatics know-how, you don't need to have the computer power to do it. And so this was really our main idea in building the database like this. You can see that it is actually encompassing a wide range of samples. We have created an ontology for our samples that is based on the Earth Microbiome Project ontology with some slight modifications that we think make more sense or a bit more modern in terms of how research has evolved in the past 8 years. Most of our data belong to hosts, and those can be plants, animals, or even, SAR. So the base of the tree, the eukaryotic tree of life, algae, and so. We have mineral, and this includes soils and sediments. We have water, and this is fresh water and seawater primarily. And we have a place to store random eccentric samples in our other category. So we have added to each study for each data point human curated metadata. And we have focused on providing data in a way that facilitates the inclusion of microbes in syntheses. Again, we bypass the need for computational power. What's really interesting now, and this is what we're basically focusing our first paper on, is this universal species definition within the database. And this is something that's entirely new, and I'm happy to discuss it. But basically, because there is no biological species definition for bacteria, we usually use an operational species definition, and an ASV is one type of operational species definition, but these depend on the bioinformatics parameters used. Because different practitioners use slightly different parameters in their bioinformatics processing, this means that the species are not comparable between the data sets. And by reprocessing all of the data together in a very specific manner, we actually have universal species. And that also brings microbes much closer to our classical way of studying ecology. And we are currently integrating with GBIF. So the whole data set will be absorbed by GBIF with versioning. MiCoDa version 2 has broken GBIF. They're trying to figure that out after the summer. We are very proud of that. Yes. What we saw from these maps is that obviously there's a big data vacuum in the global south, and this is where the Datathons come in. So the idea of the Datathons kind of summarizes a lot of the ideas I've been talking about until now, but the point is that we want to encourage synthesis of local data by local researchers, right? We don't want only researchers from the global north creating these global syntheses. We also don't think necessarily that syntheses need to be global. So with the example I gave you earlier with the agricultural inputs, there is a lot of space for analyses of local agricultural inputs, maybe at a continental level. And these are just not being done because of a set of reasons that I will discuss now. So we've now created a data set that is accessible, reusable, and homogeneous, but we lack the local data. And that is one of the main aims of the Datathon. And we want to bring people together in order to create this data set, improve the quality of the data and metadata that is archived during the Datathon and into the future, and to strengthen local collaborative networks. So the Datathon is a 3-day event. To date, it has been held 4 times, 1, 2, 3? Yes, 3 times. I'm currently planning the 4th. And it's to date been a virtual event that lasts 3 days. The first day aims to inspire, the second day aims to train, and the third day aims to collaborate. So the first day is really targeting researchers from all career stages who might never have considered the second life that their data might have. What we do is we invite speakers for a symposium, and we specifically invite speakers who have already performed data syntheses to talk about how they've reused data, what kind of studies they've done, what kind of questions they've answered. And what we found is that participants find this to be extremely useful because they can now be inspired to think about their data in a way that they haven't before, right? Because oftentimes data creators don't even get a notification with when their data is used. They don't even know how their data might be used. They might read articles on Nature and say, hey, that's really nice, but that's not what I do. And so what this lecture starts to do is to shorten that distance between researchers, wherever the Datathon is happening that year, and creating a synthesis that is impactful, that is large. And so that's what the first day does. The second day aims to train researchers in how to archive their data and to do it simultaneously and to do it with assistance. So on the second day, we open a Slack channel. We've created training materials, so we have them in English and in Spanish right now. I think we're going to be developing one in Chinese soon. The second day, researchers have a very short lecture. They then open these guides. They are step-by-step guides with a lot of images. And what's a little bit different than the kind of guidance you might get from the portal itself is, first of all, we've included a lot of images and arrows, so it's very intuitive. But the other thing that we've done is we've created our own metadata sheets and given them to all participants. And those metadata sheets satisfy all of the mixes standards, but they also are formatted for inclusion in MiCoDa. And this is very useful because it means that we can get all of the extra information that we need in order to add things to MiCoDa directly. We've added controlled vocabularies. It is not possible for researchers to pick things that are not in our list for some categories. And this allows us to really bring all of the data together much more easily. The other thing I will say is that we open the Slack channel, and in the Slack channel, people that need direct assistance get it from the organizers of the Datathon. We open it for the entire day. And our experience is that oftentimes by the end of the day, a lot of people have archived their data, and others have basically learned a couple of new things. And an example of this is rhizosphere. So rhizosphere is the microbial community that is associated with the roots of plants. It is not wrong to consider the rhizosphere part of the soil microbiome. It is also not wrong to consider it part of the host-associated microbiome. What we do in the Slack channels is arrange it so that there is consensus on all of these little things that might emerge during data archiving in a live way, so that by the end of the day, we really have a list of all of the data that has been archived. When users have already archived data, they're also invited to just send us an updated metadata sheet formatted like we requested, and this allows us to pull the data from the archives using the MiCoDa metadata. And what we offer basically is all people who deposit data for each project that they deposit, for each data set that they deposit, we allow 3 co-authors to receive credit in the final data paper that we publish about the Datathon. And there might be exceptions, of course, and that's absolutely fine. But the idea is that anyone who deposits data then becomes part of a larger data paper or data adjacent paper. And we found that this is, a really great way to bring people together. Another thing that we found is that in the global south, depending on how much funding is available for research, data sets can be extremely small. But because they come from blind spots, they are still important. But this makes people reusing data, this gives people reusing data a lot of extra work, because if you need to spend the same amount of time looking for a very small data set as for a very big one, you're going to let the smaller data set slide. Putting all of the data that we collect or in the Datathon together creates one nice place for researchers to go to get data from that region. Finally, on the 3rd day, we aim to collaborate. So we begin the day with a list of the data sets that were deposited and some basic stats. Okay, we have this number of soil samples, this number of something else. What's something unusual about this group is that we've collected really a lot of samples from this biome. And then that initiates a brainstorming session. And this leads to collaborative groups. We ask participants to self-organize over that day. And by the end of the day, the groups can present, and they create breakout groups. By the end of the day, we want each of those groups to present their findings to the rest of the participants, choose a leader, and then that leader becomes in charge of organizing regular meetings in order to move the project along. So I'm going to give you a very clear example of our first Datathon. So I am from Uruguay, and this is kind of one of the things that led me to do this. I noticed that my map had no points from my country, even though I knew that there was a lot of data there. So we organized the first Datathon as a pilot in Uruguay and Argentina in 2022. And this was done with researchers from Uruguay and Argentina. And what we really managed to do here is get almost 1,000 samples. And this was a huge surprise. The largest data set of the region until then was 80 samples. So this was huge. The data was compiled. We wrote a paper. And in this case, because it was the first Datathon, instead of writing a data paper, we wrote a description of the Datathon events. And then all participants who deposited data were co-authors. And obviously, the data was made available through this publication. After that, we were able to successfully get new team members. So Clara is now a postdoc working on the Latin America Datathon, and Santiago is our data curator, and he manages our help desk. And since then, we've been organizing a series of very specific courses aimed at training these groups of researchers towards being able to complete the projects that they came up with during the brainstorming session. So the first one was models and model selection for quantitative evidence synthesis. We've held one on advanced biostats. I've taught my introduction to bioinformatics several times. And the idea, and we are also sending out monthly newsletters for the past year. And the idea is to build a network, right? So researchers can send our help desk any opportunities that they want to advertise, whether it's a conference that they're organizing, whether it's a position that opened up or a scholarship, and we advertise it. We also advertise the courses there. And so this has been really nice. The following year, we decided to go much bigger. So we organized the Datathon in Latin America. Same structure, but look at what we did with a map. So this, I think, is a map that we should be extremely proud of. It has really, it's the largest collection of microbiome data in Latin America by orders of magnitude. And yeah, it was, I would say it was a success. It led to these collaborative networks strengthening. So the first year, there wasn't a lot of action, but by the time the second year rolled around, the Argentinians and Uruguayans were still involved in a much more central way, and they were more comfortable really organizing and leading these synthesis groups. These synthesis groups will be coming to IDIV in September for their final meeting. So the leaders of each will come, and we will finalize all of the analyses that we've been working on for the past year and a half. And hopefully that will lead to some very nice publications that will really change the field, because to date, there haven't been a lot of syntheses by Latin American researchers. So you can really see how we can change a map and shed light on these biodiversity blind spots through community action. And I think this is pretty great. For the Datathon Africa, we also had a lot of participants. What you see here in the map is our network of organizers. So we have a very solid network of organizers. We've now started our trainings. They begin in October of this year. And we just surpassed 1,000 samples from the region. We are still open to data collection. And I also want to share this. So if you look at who's participated, we're really trying to work on our outreach a bit more, but we have a pretty solid rate of about 30% of the data sets that are collected weren't archived before. They're just archived as part of the Datathon. So this is really nice. And we also have researchers from all sorts of career stages joining. And this is also really interesting. We've tried to build an event that is interesting on different days, potentially to different types of researchers. And we have a plan for continuing the Datathons. So this year in 2025, we are going to be organizing the Datathon China. Actually, we are probably going to organize it for in June of 2026. So next year will be a busy year because we're organizing two. And then at the end of 2026, we will also organize the Polar Datathon. In 2026, in 2027, we're gonna organize the Eastern Europe and Balkans Datathon. And so in this way, we really hope to start filling in the gaps. What you see here is MiCoDa version 2. And so I kind of wanna finish this presentation with talking a little bit about the plans going forward for MiCoDa. You can see MiCoDa version 2. A lot of the additions to this have been large compendia of data sets that were added. But we want to kind of automate the process going forward. And in order to keep this species definition that I mentioned earlier stable and to be able to do this in a scalable manner, we've started working on the software end of things. So we've now built HVR Locator, which is a workflow for identifying where in the 16S region a sequence was targeting from the data itself without having to go to the metadata. We've also found that there's a consistent error rate. You can see down here in pink. That the researchers give one region, but actually when we look at the sequence, it's a different region. And so to really clean up our species definitions, we now started using HVR Locator. In the future, what we're going to be working on is doing part of the metadata extraction with machine learning. And instead of doing the entire curation manually, we're just going to have our data curators confirm that the excerpts that the AI, the large language model pulled out are the correct excerpts to support the metadata that it collected. And with that, I would like to thank you for your attention.